Right. Hi, everyone. Thank you so much for joining us here today. It is very nice to see people in real life. I was saying earlier, I think this might be my first live presentation I've done since the pandemic, I think, because they're all virtual now. Yeah, but anyways, I'm Julia Publicover. I'm a director for translational research and innovation at UHN. I wear a few different hats. That's why people kind of point to me. But today I'm going to introduce you to QIPCM, which is our imaging core lab. And of course, we're also part of the OICR's adaptive oncology program. So QIPCM, it stands for Quantitative Imaging in Personalized Cancer Medicine, and it's an imaging core lab, and we provide lots of different services for multi-center clinical trials in order for them to collect top quality imaging data and have the ability to collaborate effectively across multiple different institutions globally. So the primary aims of the program include a centralized data platform to anonymize and centralize the imaging data across all of these different institutions and to allow the researchers to securely access the data from anywhere. We also have a number of software tools for research use. And then beyond the data platform, we have a suite of procedures and tests to ensure that the study is collecting top quality and standardized data across all the sites. And beyond that, we've got our support team as well. And this is our team. Our PI is Dr. Brad Wouters. He's UHN's EVP of Science and Research. And our team consists of research associates, physicists, software developers, and business analysts. Later today, Jenny Lee and Brandon Driscoll, who are on my left, you're right. They're going to be doing our virtual lab tours, and they're going to actually give live demos of some of the tools that I'm going to talk about today. So QIPCM, we were created about 10 years ago now, and the number of studies that we've supported has been growing year over year. So the light blue line on the top of this graph that you can see here, that's the total number of studies that are on our platform right now. So right now we're just over 80 studies. And then the orange line below that, that's the number of unique hospitals that we've worked with. And so right now we're at about 65, I believe, of new hospitals. Then a couple of years ago, we saw increased demand in support for AI studies. And so we've started tracking those as well. That's the green line towards the bottom. And we've got about 15 AI studies on the platform right now. Then the lines that are labeled patients, exams, and images, these are all different measures of the amount of data that we've ingested. And the sharp spikes are all driven by these AI studies. So this diagram shows the general workflow for the data platform. On the left, we've got a hospital, can be anywhere in the world, and it's participating in an imaging study. So we'll work with that site to set up our image anonymization and transfer pipeline. So this is called CTP, or Clinical Trials Processor. And it's a software that was developed by the RSNA, and it's used by many clinical trials globally. So we'll work with the site to set that up, and then the data will be automatically transferred to our central infrastructure, which sits within the UHN firewall. And then as the data comes in, it arrives in our QC staging area, where we perform both a manual QA check as well as automatic quality assurance. And there's a long list of different things that we check at this stage to ensure that we're getting top quality data. So these are things like ensuring that there's no residual PHI, ensuring there's no missing images, that the site has followed the right imaging protocol. And we can do some protocol-specific checks as well, things like checking the radiopharmaceutical injection time and things like that. So QA is performed on the data prior to it being released to our PACS and then made available to investigators. So once QA is done and it comes into our PACS, so a PACS, it stands for Picture Archiving and Communication Systems, and it's a method for storing radiological images. Our PACS is partitioned so that only approved investigators can access the data for specified and approved studies. Then if you look on the right-hand side, these are investigators. And again, the investigators can be anywhere in the world. and they can log in to access the data. So long as they have the correct permissions in order to access that data. So the method for this is that they log in through two-factor authentication, and then they can analyze or review the images in one of two ways. So it can be done first through the web viewer, which is a lightweight way to view and read images. And then there's also a virtual machine, and this is if somebody needs more compute power or different softwares in order to analyze the images. And both of these methods are going to be demonstrated during our lab virtual tours later on today. And then throughout all of this, the data access is audit logged quite thoroughly. So as I mentioned, we provide two ways to access the data once they're in our centralized repository. So first, we can spin up a virtual machine or a VM. And so the VM, it behaves just like a PC. It can be given resources like RAM or storage, just like a PC can. And on the VMs, we can install an assortment of softwares that can be used by the researchers. So some of the common softwares that are used are MIM or Python, 3D Slicer, MATLAB, and any of these can be installed at the user's request. And we can also install many other different softwares if there's a specialty software that the user needs. And then through the VM, they can access their data. Oh, and we can also provide access to the local compute cluster if they need GPU access as well. So that's HPC for health. So the second way that we provide access is through a DICOM web viewer, and this is a lightweight and cost-effective way for them to view the images. So our web viewer is built on a program called OHIF, or Open Health Imaging Foundation. We have Ali Reza here today with us from OHIF. And it's open source web-based platform, and it's extensible. So we've been working with Ali Reza and the OHIF team to build new features. And this viewer allows our users to view the images on any web browser, and it's got many of the tools that they need for annotation and review. So the way it works is it renders the images on the client or the user side. So the pixels are streamed to them, just kind of like watching a movie on Netflix. So the data files are not downloaded to the user's computer. There's no caching of temporary files. So it is all encrypted as well as the data is transferred. So beyond the data platform, we've got a number of services, including scanner QA and imaging protocol development. And then during the demo today, Jenny and Brandon are going to walk you through how sites will transfer the data through CTP, to go through the quality assurance process, and then how investigators can log in to review their images. We also have in-house expertise in dosimetry for radiotheranostics, including SPECT-CT scanner calibration for the dosimetry. We can support radiotherapy plan collection and remote review. And we've got services for AI and radiomic studies, including data curation and data quality checks, as well as feature extraction for radiomics. So our radiomics pipeline, it integrates our packs with a Python compute environment together with a user interface within our broader toolbox to compute radiomics using PyRadiomics. And our AI sandbox will all be demonstrated today as well during the virtual tours. So we've taken many measures to ensure that security and privacy are maintained throughout all of this. The data is transferred from sites through a secure pipeline, and it always remains behind the UHN firewall, and it can't be downloaded to any remote computers. Two-factor authentication is always used for login, and audit logs are always maintained. All PHI is removed from the original data prior to it leaving the originating site and being transferred to us. And our de-identification tools are fully customizable to the needs of the trial. So a key security feature is that the data access is only granted to individuals named by the PI or sponsor, and the individuals only have access to the specific data that was specified by the PI or the sponsor. And then this access can be granted or it can be revoked at any time as needed. And of course, all applicable regulatory regulations are adhered to. So we've worked with many sites in Ontario, across Canada, and really globally, and we continue to keep adding more. The table on the left-hand side here shows the breakdown of how many studies a site is either leading, so that means that the lead PI resides at that institution, or the number of studies that that site is participating in. So on the right, we can see we've worked with UHN and London quite a bit. They've got lots of studies with us. And then we've got many sites across Canada that are participating in those. Outside of Ontario, across Canada, we've got 45 sites that are participating in different studies. And internationally, we have an additional 28 sites that work with us. And we've got seven industry studies with us. And so on the right-hand side, your left-hand side, are the sites that we work with. So looking forward, in addition to our centralized data platform, we're working towards building a federated data platform as well. So a federated data platform is where the imaging data can be viewed and analyzed by external users while the data remains at each institution. So we would not centralize it. The data would remain at the originating hospitals. So we've conducted a pilot project with BC Cancer where we connected our web viewer, the OHIF web viewer, to two data sources, one located at QIPCM here in Toronto, and then the other one at BC Cancer out in Vancouver. And then the imaging data infrastructure was hosted on premise at each institution. So this project, it's evolving still as we're developing our use cases for it and connecting with different stakeholders, one of which is the Terry Fox DHDP. And they're building a larger federated data network. And we're hoping that we can leverage some of what they're building in order to connect to a diverse set of data beyond just the imaging. So now I'm going to walk through just a handful of case studies which highlight how the infrastructure I just described is applied to support a variety of different clinical trials and studies. But the platform it's very versatile, right? You can use it for a lot of different things. And this is only a small sample of what we can provide. So if you've got a project in mind that I haven't demonstrated here, please don't hesitate to come and talk to me or anybody else on the team to see if it's something that we can support. So first, I'm going to discuss Patron. This is a study that is led by Dr. Cynthia Menard out of CHUM in Montreal. The long name for Patron is PSMA PET-CT guided intensification of therapy in patients at risk of advanced prostate cancer. So this is a phase 3 randomized control trial, and it's looking if PSMA PET-CT guided intensification of radiotherapy or surgery can improve cancer outcomes. So in this case, images and radiotherapy plans from almost 800 patients and across 15 different hospitals need to be anonymized and collected. The images are rated locally by the home institution's radiologist, but they also need to be reviewed by an expert central reviewer prior to randomization. And the same is true for the radiotherapy plans as well. So the way that QIPCM supports this trial is by using our customized CTP to anonymize and transfer the images from these 15 sites. Then an automated email is sent out to the different sites to confirm receipt of the data. And then after that, the images undergo automated QA, checking the image completeness and the adherence to the imaging protocol. And then a fraction of the images get manually QA'd as well to ensure there's no embedded PHI and there's no image quality issues as well. So once QA is complete, the radiologists or radiation oncologists are notified that there's a review pending for them, and at which point they're going to log into their QIPCM virtual desktop to perform their review. So the QIPCM VM, it's a standard Windows desktop, and it's hosted through Citrix Workspace, and it's contained, as I mentioned, entirely within the UHN firewall. So from here, they can load up a plan or an image set in MIM is what they're using and perform their review. So at this point, the trial has accrued all nearly 800 patients, and it went through that very quickly, and we've been hosting the data for them as they do their final closeout of the study. So I'm going to switch streams here a little bit and talk about the Octane trials. These are led by Dr. Phil Bedard, and this is also part of the adaptive oncology program here at OICR. So these are a good example of how QIPCMs worked with other teams to link our imaging database to other available clinical data. So Octane stands for Ontario-wide Cancer-Targeted Nucleic Acid Evaluation. It's actually two different studies, Octane-1 and Octane-2, and they're pan-cancer studies, and they look to provide biomarker data about a patient's cancer that may be able to help their physicians to identify new clinical trials for drug treatments that might be appropriate for the patients, and it might also guide the use of approved treatments for the benefit of patients. So Octane is kind of special because it's also looking at linking data from their studies to other healthcare databases to further collect information about the healthcare the patients are receiving, including medical tests, clinical visits, and procedures. And this also includes genomic data as well. So as in the previous cases, both the Octane-1 and Octane-2 trials are collecting imaging data from multiple different sites. However, the primary difference with Octane is that they're collecting large amounts of genomic data and tissue sample data, which is made available via another web-based solution called cBioportal, and this is managed by Dr. Trevor Pugh's team, also part of OICR. So the goal was to work with the cBioportal team to see if we could connect our imaging data with the cBioportal data as well. So for this study, we've set up the CTP anonymizer again to anonymize and centralize the imaging data. We did the QA for the data as well using our imaging toolbox. And then we worked with the cBioportal team to create a link between their platform and our web viewer to allow researchers to view imaging data alongside all the other clinical data that's been collected. So as part of this study, they're also planning on extracting radiomics feature sets, and this is going to be made available in cBioportal as well. And so we're working closely with the team to extract the radiomics features. So this is just a teaser because if you'd like to see more about how we've linked to the data sets in cBioportal, we're going to have a demo of that during the virtual tours. And then the final case study I'm going to discuss involves supporting the development of AI-based tools and models that are being developed both by academics as well as industry partners through the use of our AI sandbox. So there's many researchers as well as companies that are very interested in building and training AI and machine learning algorithms using the valuable imaging data that's collected all the time along with the clinical data. So unfortunately, questions of data ownership, patient privacy, and intellectual property make it very, very difficult to export these large data sets to external partners, particularly if they're industry partners. So we need to develop a solution to make this work. So our solution currently has been to work closely with these industry and academic partners to obtain the approvals needed, and then we anonymize and collect the data from the clinical packs, which we then make available to the researchers and to industry through our QIPCM framework so that the data never actually leaves the hospital itself. And so if you actually want to see this one as well, we're going to do a demo as well. So you can see how the data is anonymized, where we store it, and how it can be used while it still remains within the hospital. And so that is really my last slide. If you want to talk about any projects that you have in mind, please reach out. We've got a web form through our website, but you can also just email me, anybody else on the team, and we're happy to talk about what your projects might be. Thank you very much.